Okay, so today's video is going to focus on going from a gene to a protein. So a gene, let's get some vocabulary real quick. This is just a section of DNA that codes for one protein. So if we remember, our DNA is in big, long strands. And so those strands contain genes. So for instance, if I look at this picture of DNA over on the left here, the gene is this portion here, or this portion here, or this portion here. If I look up here on the right, right, like maybe this is a gene, or from here to here is a gene, right? So there's thousands of genes on one strand of DNA. So it's just the section of DNA that codes for one particular protein. So we're going to look at how we're going to actually make that protein, because while the DNA carries the genetic information, the proteins are the ones that are actually carrying out the work and doing the jobs. Okay, so in biology, we have what's called the central dogma. And the central dogma is how the information within a cell flows. So the start of that would be at DNA. So there's the actual genetic information. From the DNA, the information will flow to the RNA, to the protein, and then it will be manifested within the organism, showing up as a particular trait. So again, while the DNA carries all of the information, we don't let's say, for example, let's talk eye color, right? So eye color we know is determined by the DNA that we got, but the actual eye color we see is due to proteins. DNA had the instructions for that particular set of proteins. Those proteins were made, we got brown eyes, blue eyes, green eyes, whatever. Okay, so that's our central dogma, DNA to RNA to a protein. Okay, so in the cell, how that will look, in particular, we're talking eukaryotes here. So eukaryotic cells in the nucleus. So first we've got DNA to RNA. That process is going to be called transcription. It's going to happen in the nucleus because DNA can't leave the nucleus. So the DNA to RNA is transcription occurring here in the nucleus. Then that messenger RNA, that's what the M stands for. We'll look at different types of RNA in just a minute. That messenger RNA can leave the nucleus. If you think back to when we talked about nucleus and the structure of the cell and the nucleus having those nuclear pores, DNA is too big because it's double-stranded, so it can't leave the nucleus. But the messenger RNA is single-stranded, so it is able to leave the nucleus with the information. It's small enough to fit through those nuclear pores. So the messenger RNA heads out into the cytoplasm. Out in the cytoplasm is where translation will occur. Translation is the process of taking that messenger RNA to a protein. This is where ribosomes come into play. When we say that ribosomes make proteins, ribosomes help convert that messenger RNA into the protein. And then again, that protein will show up as the trait. So let's look at each of these in a little bit more detail. Okay, so transcription is our DNA to messenger RNA. Remember, we said that this is going to occur in the nucleus because the DNA is located in the nucleus and is unable to leave. Okay, so transcription, DNA to messenger RNA. So let's talk about RNA a little bit here before we go any further. So RNA, its monomer is going to be the nucleotide, just like it was for DNA. Remember, a nucleotide contained a sugar, a phosphate, and a nitrogen base. So RNA is going to be a little bit different because while it contains a sugar, this time the sugar is ribose. Remember, for DNA, the sugar was deoxyribose. So the sugar in RNA is ribose. The phosphate is the same. The nitrogen bases, though, are a little bit different. Remember, for RNA, we have uracil, adenine, cytosine, and guanine. So no thymine in RNA. So as you get pictures from now on, you need to start paying very close attention to, is this RNA? Is this DNA? So is it one strand? Does it have U's? Does it have a ribose? Okay, it's RNA. Is it double-stranded? It has thymine, it has deoxyribose. Okay, that's DNA. Okay, and then I also need to be looking at the process. Am I making a copy of DNA or am I going DNA to RNA? Because that will dictate, again, whether that uracil is present or a thymine. RNA also has 3 different types, which DNA does not. DNA only has the one type, right? So it has messenger RNA, which we've been mentioning already, that it's important in transcription, getting the information out of the nucleus. It has tRNA, which is transfer RNA, which will be important when we talk about translation. And then it has rRNA, which is ribosomal RNA. We don't talk much about rRNA, the ribosomal RNA. Basically, rRNA is RNA that you're going to find in the ribosome. And so it's going to be part of the ribosome, which will be important again in translation, but it doesn't carry information or anything like the messenger RNA and the transfer RNA does. So again, be sure you're able to compare DNA and RNA. That will be an important skill. Okay, so let's look a little bit closer at this process of transcription. So transcription, again, is DNA to RNA. So you'll see I have my DNA strand in this diagram here. Okay, I have 2 parts of it. I have the coding strand and I have the template strand. You'll notice the template is what is providing the blueprint to actually make the mRNA. This green strand here is my mRNA. You'll see that it's single-stranded. If we labeled this, I'd have uracil, not thymine. And then the last thing we have in this image here is this RNA polymerase. ACE should be cluing you in here that this is an enzyme. So like all chemical reactions, transcription, or that DNA to mRNA process, is controlled by enzymes. RNA polymerase in particular is going to help build RNA. Poly means many, right? So I'm building many RNAs here. So this enzyme is an important enzyme in regulating the process of transcription. So this messenger RNA is going to be made by using the template strand. Let's take a look here a little bit if we labeled these. So let's say that, let's label this DNA. So I have C, G, A, G, T, A, C. So on my other side of the DNA, I'm going to go up here so you can read it better, right? I have a G, a C, a T, C, A, T. Oops, I went too far over. Let me shift all those down by 1 real quick. Okay, let's actually line them up here. So G, C, T, C, A, T. So those 2 DNA strands, you'd see how they would complementary base pair to one another, right? Remember, they aren't going to directly match. They use this complementary base pairing to form their strands. When I unwind this DNA, that's also controlled by an enzyme, helicase, right? The helix, I'm going to unwind the DNA helix using the enzyme helicase. When I unwind that DNA, I can now make copies for my messenger RNA. So this C would have to bond to a G on the messenger RNA. You know what, again, I'm going to go above so you can see it better. So the C, that would be a G, G to C, A, but because I'm making RNA, A goes with U. Remember, U takes the place of T. U does not bond with T. It takes the place of T. Okay, remember we had our pyrimidines are cut from purines. So pyrimidines are cytosine, uracil, thymine. So uracil and thymine are both pyrimidines. 2 pyrimidines can't bond to one another, right? So that A goes with a U because I'm making RNA. G with a C. Now I have a T. T does not bond with U. That would be 2 pyrimidines. T bonds with A. A with a U, C with a G. So now I have this complementary messenger RNA strand that if you look and compare this coding strand of the DNA to the messenger RNA strand, they are the same. Okay, so I use my template strand to build my messenger RNA strand, and then that messenger RNA strand is going to leave the nucleus and take that information out into the cytoplasm. Okay, so we already talked about this, so just a few reminders, right? Here's our RNA polymerase enzyme that is building the RNA, attaching those RNA nucleotides. Remember, we build in the 5 to 3' direction. We talked briefly, we talked about DNA. We talked about the carbons, numbering the carbons. So this is carbon number 5 over here on the left, carbon number 3 over here on the right. Okay, I'm complementary base pairing. So let's go ahead and just practice and finish out this messenger RNA strand that we would be making. So it's already showed us here that the T would go with the A, the U with the A, C, A, G. Pause for a second. See if you can go ahead and finish that strand out and then check your work. Okay, so hopefully you gave it a try on your own. So the A would go with a U, T with an A, C to G, G to C, T to A, never to U, A to U, C to G, C to G, G to C, T to A. That's what your rest of your strand should have looked like. Okay, so one of the things you also may be asked to do in addition to doing transcription, so in addition to taking that DNA and writing what the mRNA strand would be, sometimes you're just asked to pick it out from a picture. So there's a wide variety of pictures you could see. One of the key things that you need to keep in mind, okay, is that you're gonna see DNA and messenger RNA in the picture, meaning you're gonna see a double strand and a single strand in the picture for it to be showing you transcription. So you can see in this top image here, here's my mRNA being made coming out the top, and then the DNA running through the middle here. This picture on the bottom left that's unlabeled, it's not labeled at all, but I can see that the purple here is single-stranded. So that gives me a clue that that's mRNA, and then I have my double-stranded DNA. Same in this bottom picture to the right, just this time I have my mRNA, my single strand coming out from the bottom. Okay, so let's shift gears a little bit to translation. So translation is the second portion of the central dogma, taking that mRNA and completely turning it now into a protein. This is going to take place out in the cytoplasm using those ribosomes. So the DNA is no longer part of this anymore. This is taking mRNA to a protein. That's the translation portion. Okay, so let's look a little bit closer at how this is going to do this. So we already did DNA to mRNA, right? That's my transcription. And then the organism or the cell is going to take those mRNA strands and they're going to clump them into groups of 3 to help do translation. You can see groups of 3, they color coded them here. Your papers, you may need to do this the first few times you do this, you may need to block them off into the groups of 3. Those groups of 3 are called a codon. So this is groups of 3 nitrogen bases or 3 nucleotides. Those groups of 3 are called a codon, and each codon codes for 1 amino acid. And if you remember back to proteins, the monomer of proteins are amino acids. And so for the monomer of protein being an amino acid, I need to link up a whole bunch of amino acids together to actually make a protein. So each tiny little group of 3 codes for 1 amino acid. So then the big long strand of mRNA can code for 1 protein. Okay, so let's look now how we use those codons to actually determine the amino acid that it's coding for. So we're going to use what we call a codon chart. Codon chart always has to be used with messenger RNA. You can't use it with DNA. So the codon chart, let's make a little mRNA strand here. Okay, so remember we break these into threes. And then I take my first codon, which is CUG. So remember this little branch here is a codon. And you'll see I read my chart left, top, right. So my first base is over here on the left. So I have a C. So I go find the chunk that has C's. So I know I'm in this row right here. I'm not using any of the rest of my chart because my first base was a C. My second one is a U. So now I come up to the top. Second base is on the top. I find the U. So now I know I'm in this first column here. So now I know I'm in this box. I've got it narrowed down to this box right here. It actually doesn't matter what the third base is because all four of those, all the CU possibilities are leucine. However, if it did matter, hopefully I made one up where it does matter. Okay, I didn't. If it did matter, we'd come over to the far right where it says third base. I'd find the G, so this one right here. I know it's that particular G because I'm in the C row, which leaves me with this one right here, CUG, leucine. Okay, pause for a second, try to do the next two, and then check your work. Okay, so for the next one, again, we were in the C row. And our second base this time was C. So we know we're in this box right here. And again, it didn't matter, but that one would be proline. For the third one, we needed to move up to the first row for U, G. So we're in this section right here. And it did matter what the third base was. The third base was the A. So that puts us right here. That literally does mean stop. Okay, if you get a stop codon, that is how the cell knows it's reached the end of making that particular protein. If we do mutations later and you get a stop in the middle, you don't keep going. It literally means stop. This is the end of the protein. It can be released and finish folding and done whatever it needs, go do whatever it needs to do. You may also see a circular codon chart. Circular codon charts work inside to out. So for example, let's say you had A, U, C. So I would start in the middle at the A, and then I just follow to the U, to the C, and I end up with isoleucine. So the circular codon chart is sometimes easier, I think, to use. I can't guarantee you'll always be given a circular codon chart though. So you need to be able to use both. Okay, so how do the amino acids actually get there? Now the messenger RNA is telling the cell, the ribosome, which amino acid needs to be placed there, but the amino acid doesn't move there, right? The amino acid is brought there by the transfer RNA. So remember, tRNA is transfer RNA. It's going to carry or transfer amino acids. So you can see I have my codons here. The tRNA, because it's made of RNA, it can complementary base pair. See how I have an A to U, U to A, G to C. And this amino acid, this tRNA carries the amino acid methionine. tRNA is amino acid specific, meaning that tRNA only carries one amino acid. It's reusable, it drops it off, but it only carries one. So that way, the tRNA that has UAC on the end can only bind to AUG, can only carry methionine. Therefore, that guarantees that methionine is going to end up in that first spot. Okay, so for example, the very last one, the tRNA would have an anticodon of CGG, right? These are my codons because I complementary base pair, right? Anti is against. There's my anticodon. And so on the other end of this tRNA, I'm going to carry the specific amino acid that goes with that. Remember, it's not going to be CGG. I use a codon chart. So I would go to my chart. I would find GCC. And that particular corresponding amino acid would be on the other end of the tRNA. And it would carry an alanine on it. Okay, and that's how you can determine that, but that's how those amino acids will get there. So tRNA or transfer RNA is one of the things that can really help make a translation picture stand out from a transcription picture. So a lot of times tRNA is depicted either in this horseshoe manner, like we kind of drew on the other page. Okay, so it'll have the bonding sites, the anticodons on one end, and it will carry the amino acid on the other end. Sometimes it is written, as you can see, this kind of cylinder like leaning, where again, it will have an anticodon on one end and it will carry an amino acid on the other end. Remember, they're reusable, they're not consumed, and they don't become a part of the protein. They drop the amino acid off, and then they'll go back and pick up, like for the example, we used the other one, the alanine one, it would drop that alanine off, and then it would go back and pick up another alanine. Okay, so ribosomes are going to help put the finishing touches on this and really help. Ribosomes are really what's going to help the amino acids link together. So the ribosome is made out of rRNA or ribosomal RNA. It's made of these two parts, this large and the small subunit. So what will happen is the mRNA will thread between the two units, and then the tRNA will bring amino acids over. So for instance, I have a triangle amino acid here, and then this amino acid on this one. Let's make this one a square. And then this one is leaving, and it dropped off a circle one, and it's going to go pick up another circle, right? And so the ribosome helps the tRNA, helps the peptide bond. Remember, a synonym for proteins is polypeptide. Helps the peptide bond form between the different amino acids, and then helps the ribosome send those transfer RNAs on their way to go pick up new ones. So that eventually I end up with just the string of amino acids or the polypeptide that's held together with those peptide bonds. So looking at some different pictures of translation, the things with translation that you would need to be able to do mainly is read the codon chart. Okay, so determine the order of the amino acids, and again, pick out pictures of it like with transcription. So with translation, for pictures, keep an eye out for the tRNA. You're only going to see that on translation pictures. So you'll see multiple types of RNA, tRNA, mRNA, and you'll see either the amino acids or the protein chain, the polypeptide chain as a whole. You can see on the one on the top left, here's my protein forming as I'm attaching amino acids to it that are being brought over by those transfer RNA. Same again, my polypeptide chain is forming. Those peptide bonds are being formed between the amino acids, linking them together as those transfer RNAs drop them off and then move on to pick up new ones. Again, at the bottom, just a slightly different format for it. Okay, so like always, not losing sight of the big picture here. So I've got transcription happening inside the nucleus, DNA to mRNA. mRNA leaves the nucleus, goes out to the cytoplasm for translation. And you can see I'm zooming in, right? Here's my codons. Those codons are telling the tRNA which amino acid, right? They're bringing over those amino acids, which amino acid to bring over. The ribosome is helping form those peptide bonds between the amino acids. So I end up with this long chain of amino acids, which becomes a polypeptide folded into the particular protein, which will then actually show up as the different traits and the different characteristics. Okay, like always, any questions, make note of them in your notes, write them down. Don't just star stuff because you might not remember what it was that you wanted to actually ask about. jot the actual question down so we can clear anything up before we start modeling and practicing with this in class.